title of the talk is DNA-based timer tracking for forensic and legal enforcement. The sub-theme is horticulture and forestry. The talk is going to delivered by Dr. Suma Arun Dev. Dr. Suma Arun Dev is a principal scientist and head of the division of forest genetics and biotechnology at the KSCSTE, Kerala Forest Research Institute, Pichi. She earned her PhD from Cochin University of Science and Technology in the year 2003 and later held a DPT National Postdoctoral Fellowship at the Indian Institute of Science, Bangalore. Since joining KFRI in 2010, she had several national and international research projects. Her major contributions include in the development of DNA reference and barcode databases for teak, sandalwood, palms, commercial bamboos, Ayurvedic raw drugs and tropical timber species. She also played a key role in establishing India's first Center for DNA barcoding and timber forensic at KFRI. Dr. Suma has published over 100 research papers and has received several honors including Kerala Science Congress Young Scientist Award, Professor Abraham Gold Medal, CSIR and Gate Fellowship, and International Travel Fellowships. She served as Associate Editor of Tree Genetics and Genomics and as an editorial board member of PMC Biotechnology. Her research interests focus on forest and conservation genomics, DNA barcoding, and timber forensics. With this introduction, I request the Dr. Suma Arun Dev to deliver the talk.
So, thank you.
Over to Madam.
So, thank you, Professor, for the very warm introduction. In fact, it's nice listening to you, sir, like after such a long time. So, respected professors of the university and dear students. So, today I'll be speaking on the potential of DNA tools for timber forensic applications and also for the legal enforcement. So, before I begin the topic, it's not going. So I'll briefly introduce you about our institute. I'm from Kerala Forest. I think none of you must have visited KFRA, Kerala Forest Research Institute. It is at Thrissur, located at Thrissur, 23 kilometers away from the Thrissur city. It is coming under KCST, Government of Kerala. And it is situated just adjacent to the Pichi Valani Wildlife Sanctuary. And we have a team of some 27 scientists across 9 research divisions, and in addition to the main campus at Pichi, we have a sub-center at Nilambur. You must have visited, like there is a bioresource park and the unique teak museum. The world's only one teak museum is housed at this particular sub-center at Nilambur, and we have one more sub-center that is in Thrissur itself, where we do the commercial production of plantlets through plant tissue culture. So there are 2 sub-centers in addition to the main campus, and we have field research stations in almost all the diverse, 4 field research stations in diverse forest types. So, and in addition to that, we have many live collections, like we have an arboretum where we have the live collections of the tropical tree species, and we have the bamboo situm, of which we have a collection of 75 species that is also as part of KFRA, and we have a palmatum where we have have diverse collections of palms across the world. And then we have orchidarium, we have fernarium, and we have a butterfly garden, and we have medicinal plant garden, and variety of museums, like we have a soil museum where we have the collection of soil monoliths from diverse forest types, and we have wildlife museum where we have the specimen collections of across all genera of wildlife, and we have also have a wood museum. We have a that is known as silarium, and so that in 2023 at KFRI we have established the Center for DNA, for which I will be talking mostly restricted to this center. So we have established this center in 2023. In fact, the first suggestion that was come from Dr. G.M. Nair sir, when he came for my first assessment, he suggested me like, Suma, why don't you start a Center for DNA barcoding? That was long back, I think in 2013, that point of time, but it took this much years to build that confidence to start a center like this. So this is the first of its kind of facility in India which provides DNA evidences to the judiciary. Like you have the human forensics, this is for the, whenever there is a timber theft offense is happening in the country, we do submit DNA evidence to the judiciary so that they can easily convict the illegal traders. Because earlier the scenario was that they don't have any, because the court, everything works on based on the evidences, right? So earlier there were no scientific evidence in front of the court, and then most of these illegal traders were scot-free without going through the the conviction. So now that we are in a better position and we are able to provide DNA evidences in timber theft offenses, and so this center, this was actually established with the financial support from Kerala Forest Department. And whenever there is a timber offense is happening, along with the investigation team, when the case is, criminal offense is registered, along with the investigation team, like we also used to visit the field and we do the confiscated sample collection and we submit the court, and from the court it will be coming to us, and for which we submit the evidence. So now that Kerala Forest Department has made, because in the country we don't have any facility like that, so now that the Kerala Forest Department, they are our major stakeholder, so they made recommendations to the government. So the center is in the process of getting approval from the government as a nodal center for timber forensics. So the paper has been already, the proposal has been forwarded to government with positive recommendations from the head of Forest Force. So why do we need such a center in our country? We know like the illegal timber offenses are always on the rise. So like, see, even though like in the country we set up a target like by 2030 our forest cover. I don't know how many of you are from forestry side, but I will try to simplify as easy as possible so that all of us would be able to follow this. So we know like we have only 27.15% of forest cover in India, and we have set up a target by 2030 the forest cover will be increased to 33%. So in spite of all these things, like we are, over the 3 years, we can see that over 11 lakh trees has been felled across 17 states. So that much rampant the illegal felling of trees. So all this shows the different types of timber trees. We know the highly priced timber tree species like teak, sandalwood, red sandals. Most of the days we can read in the newspapers about these timber felling offenses that is happening in the country, in our state especially. And when it comes, see, in addition to the felling that is happening within the country, we have the cross-border, across countries also illegal trade is happening. See, India, we have many conservation acts and policies in place, but when it comes to enforcement, like it's very weak. So as a result of this, you see like India has become one of the third favorite destination for the illegally imported wood. So wherever the illegal felling in other countries are happening, that wood is coming to our country because we have a huge domestic demand. So that much weak is our legal enforcement system is. And especially like we know like in Myanmar, our neighboring country, like earlier it was known as Burma, in Myanmar like there is a complete extraction ban. But the teak wood from Myanmar, that is one of the famous, world famous teak wood for many applications including shipbuilding, furniture and all those things. But there is a complete extraction, logging extraction of teak wood in Myanmar. But we are sharing a border, like in the state Manipur, we are sharing a border with Myanmar, so that illegally smuggled timber logs are coming across the border to India, and from here it is exporting as Indian teak wood, but it is actually Burmese teak wood. So that much, so across borders also these illegally logged woods are smuggled, but we don't have any enforcement system which can actually identify, okay, this is from an illegal supply chain that is coming. So that we have to strengthen, so that is the very purpose of establishing centers like this. So I will just briefly touch upon the major key factors or the drivers for this illegal felling. As all of us know, like even though like we have this much forest resources in the country, we are still facing a deficiency in terms of timber production, because we have a huge domestic demand, like especially in our state, without wood nothing is possible. So we have a huge domestic demand, and there is a disparity in demand-supply ratio of wood. And we know, like, we have many conservation laws, like 1980 Forest Conservation Act, National Forest Policy, and we have also a complete extraction ban that is imposed by the Supreme Court of India. But in spite of all these, violations of all these policies are happening, again because, so that is one of the important factor, that again because especially in a country with very limited means to implement the laws, most of these timber crimes often go unnoticed because we don't have evidence, scientific evidence before the court to prove that, okay, this particular timber log, seized log is originating one particular source. So far we were not having any scientific evidence. And the second important factor is, like, whenever we have a timber crime, like in most of the countries, most of the international agencies, they have already implemented advanced scientific tools for timber authentication. But in our country, it is still like the traditional tools like anatomy and chemical tools like chromatography is being implemented. So with anatomy, like, you may not be able to confirm the source of origin, or you may not be able to identify the timber species. So that is again one of the drawbacks. So this has actually accelerated the timber theft offenses. So this is like the situation demands, like, we need to have an integrated advanced molecular tool approach by which you should be able to combat illegal felling. So you know, like the UNODC and ITTO, these are some of the international agencies which has already recommended DNA tools for timber forensic applications. So what are the DNA tools? So in this, the subsequent slides, I will highlight, like, what are the DNA tools and the chemical tools that is being recommended by the global agency, so that just like, you know, like, you are quite familiar with the human forensics, like where you do the paternity testing, where you do the STR profiling. But here in timber forensics, additionally, you need a tool, because all the human beings belong to single species. So whenever there is a crime is happening, you don't have to confirm the authenticity of the species. But traded timbers are of different species. So when it comes to a timber log, you cannot use any taxonomic features or morphological characteristics to identify that this is a teak timber, or this is a sandalwood timber, or red sandal timber. And in some cases, even the logs will be, like, it will be just full by converting the logs into veneer wood and sawn wood. So you may not be able to identify the species of the timber. So in addition to the STR profiling that is being employed in human forensic applications, we need to have an additional tool that is said to be DNA barcode. I think you must have heard about DNA barcode. That is a species authentication tool, and all the species, all the individuals of a species will be having the same DNA barcode. So once if you have the barcode, and even if it is a small bit of seized wood, a small sample of seized wood also, you cannot use morphological characteristics for confirming the identity. In that case, sometimes in the case of sandalwood and lots of adulteration is happening, and in the powder also, you know, adulteration is happening. So in those cases, if you want to confirm whether your product you are going to purchase, whether the wood you are intend to purchase is an adulterant wood, you can use this technology where you need only very small bit of tissue and you can ensure the authenticity of the species. So DNA barcode is one of the tools that is being used for timber forensic applications. The next tool is just like your STR profiling. Like in the case of paternity testing, you must be knowing, like, if you want to identify the biologic parents, you will be comparing the STR profile. Same way, like, this is being used in timber forensics, like for like if there is is a massive felling that has happened, and you have caught the seized logs from somewhere in the state, and if you want to identify from where these seized logs originate, from which forest it must have originated. So using this STR profiling, you can make a comparison between the log and the suspected stump. So the log to stump comparison can be done just like your paternity testing, or whenever there is a criminal offense is happening where the victim's DNA profile is compared to the suspects. Just like that, you can use the log to stump comparison by using STR profile. So these two are the major DNA tools that is being used in timber forensic applications. Only these two tools are required because there are only instances where you have to, most of the samples, because after the establishment of the center in 2023, so far we have attended 49 timber theft offenses. So most of the cases you have to either authenticate the species, like which is the species, because in most cases when the trial is happening in the court, they will fool the court stating that this is some other less known timber tree species, but where the court needs some scientific evidence, so where you will prove the identity of the species and you will offer the DNA barcode sequence to the court so that the court can proceed with the prosecution. So next is like whenever there is a massive felling, like we had a very recently in 2022 we had a massive felling in Wayanad of Mutil felling of rosewood. So there also you can compare the log to stump, and we submitted DNA evidence to the court. The trial is happening in the court. So in addition to that, like whenever there are massive felling is happening, physically you may not be, or manually you may not be compare the log to stump. So in that cases, we have developed some servers mentioning about the importance of AI, importance of MLA, machine learning algorithm. So for VICA is one of the machine learning algorithm which we employ for DNA barcode identification when large number of samples are coming to us for identification. And then the other two are ASEAN POP and MicroRace are again AI tools which are used for log to stump comparison when STR profiling is happening. So in addition to DNA tools, now that we are also working on isotopic signatures, so like chemical tools, the advanced chemical tools like isotopic signatures and spectral signatures can offer an integrated approach. If your DNA tools, the process of extracting DNA, you know, like from juvenile tissue, it's very easy. But when it comes to wood, it is, you know, it's a dead tissue, right? And then many of the log seized woods must be exposed in the police station or in the timber depots of different seasons, and the samples coming to us are extremely shredded. So that is the biggest challenge. So that's why it took this many years, over 15 years of research only we were able to establish this center. The biggest challenge we faced is the DNA, getting good quality DNA which can be amenable to the downstream processing. So if the sample is degraded, definitely, DNA is degraded, definitely you cannot proceed further. So our trial and error, now that we have a technique, we have developed protocols with so many modifications, and now that we have filed a process patent also for getting this good quality. Without getting good quality, you cannot do, you must be having immense, a lot of issues facing for the PCR amplification and for the STR profiling. So now that, and additionally now that what we are doing is like in addition to these timber offenses, now that forest certification is coming in a big way. So where you can see there are different supply chains. You are going and purchasing wood from market, but this wood that is coming to market is from legal supply chain as well as from an illegal supply chain. So if you want to identify the geographic origin of the wood that is coming to us, this isotopic signatures is a very precise tool. So we are developing isotopic signature database also along with DNA barcode database and STR database, and we have like spectral signatures like dark tophomus is again a species authentication tool. So in addition to your DNA tool, you are also adding spectral signatures for species authentication, like your FTIR spectra. That will actually look into the composition of the wood. So this integrated approach can effectively offer a precise DNA evidence or a scientific evidence to the court. So in case if your DNA tools fails because of so many issues, your isotopic or your spectral signatures will work. So now that we are developing an integrated tool for combating illegal felling through the development of reference databases. So I think I will briefly explain about the DNA barcode. DNA barcode, I think many of you must have heard about, it must be there in your syllabus also, but just like you must have heard about the UPC barcode in supermarket products. In all the supermarket products, we have the black and white barcodes, right? So just like your UPC barcode, which is very specific to that supermarket product, you can have a biological barcode, that is DNA barcode, that is used for species certification. So this species certification means, like, see, you don't have to barcode the entire DNA. You have certain selected gene regions which are conserved within a species. So if you select those regions and if you develop a DNA barcode database of highly traded timber tree species across the country. So as GM sir was mentioning about the importance of developing database. Without developing database, you cannot do any of the forensics, especially not like human forensics. Human forensics, you know, like since the development of the draft genome in 2003, and you are, all the human beings are of the same species, you don't need any species authentication tool. And STRs also, like in n number of studies have been carried out to develop STR profiling of the human populations across the globe. So that database is already available, and you can have very specific STRs, right? Short tandem repeat profiling is being done in human forensics, where you have STRs which can actually differentiate individuals. If you can differentiate individuals, only those STRs can be used for paternity testing or for criminal offenses, right? So likewise, you have to go for development of very specific STRs which can actually differentiate timbers, then only you can use STRs. But the main complicated challenging thing is, like, it's not a single species of timber. Illegal felling or illegal offense is happening across almost all the traded timbers. So that was the biggest challenge for us, like we have to develop the database. So we took last 15 years for developing the reference database, DNA barcode reference database for 43 traded timber tree species in the country. That includes even imported species. When you construct a house, you can go to market and you can even buy your imported timber. Like many timbers are with very attractive aesthetic colors. Like you can have timbers. So all those timbers are also coming into market. So we developed reference database, DNA barcode reference database for 43 timber tree species which are highly traded in the country. So the strength of this DNA barcode, as I had mentioned, like you don't need, see, if a taxonomist, we are not belittling taxonomists. When you develop a DNA barcode, you need a taxonomist. Right. See, when you develop a barcode, see, I think last 3-4 projects of different sponsored project only we were able to develop this barcode. So a taxonomist was one of the associate investigators of all these projects. Why we need a taxonomist? When you develop a DNA barcode for a species, your identification should be very pakka. See, only taxonomists can go to the field and identify a species. And if you yourself, as a biotechnologist, if you yourself go into the field and identify, what will happen? You will wrongly attach a barcode to a species. So when you develop a DNA barcode database without the support of a taxonomist, you cannot develop a barcode. So with the help of the taxonomy support, Dr. Srikumar from KFRA and Dr. Sujanapal from KFRA, we developed all these reference. Once if you have the reference database, and now that when small bit of wood tissue is coming to you, you can easily identify because you have a reference database. So that is the way, like, we developed a reference database. So, and again, see, as sir was mentioning, see, when we started off DNA sequencing and barcode development, per reaction, as Dr. Jasmine must be knowing, per reaction it was like 1,500. If you have to do forward and like Sanger sequencing, if you have to do forward and reverse sequencing, you have to spend 1,500 for forward and reverse reaction. But now over the years, you know, there is a drastic technological development and the cost has come down. So now that with 350 rupees, you can develop a DNA barcode. So that much cost effective the technology is. So these are some of the strengths of DNA barcode for the species identification, and even so the most important part is the reference database creation. If you are attempting to, as part of your PG program, if you are attempting to develop, I will come to the application, the potential of DNA barcode for diverse applications. So then you will be more clear on the development. So I will briefly touch upon, in addition to timber forensics, what are the forensic applications of DNA barcode. So wildlife forensics. Any idea about wildlife forensics? We know India is one of the 12 biodiversity nations. Why? It's because we have very huge diversity of animals, right? And we have the tiger population, the elephant population, the rhino populations, and we have the only one Asiatic lion population. So even though we are, and see, most of these animals are listed in, we know there is a Wildlife Conservation Act in 1972, right? So all these animals are listed in Schedule 1, 2, 3, and 4 of Wildlife Conservation Act, and where the poaching is prohibited. So, but in spite of all this, the rampant poaching that is been happening for the illegal wildlife trade, right? So when the cooked meat is being traded, how will you identify? When the small products, you cannot use any morphological features, right? So you need some tool, molecular tool, by which you will be able to. So Wildlife Institute of India is the nodal agency in the country for doing wildlife forensics now. So they are using DNA barcode tool, your CO1 barcode. You must have heard about cytochrome oxidase 1. So CO1 barcode is the universal DNA barcode, unlike plants. It's the universal barcode for across all the animal kingdom. But for plants are more diverse, not like animals, right? So we have multiple DNA barcode regions that you have to work it out, which is the best barcode for your species of interest. Okay, so this is again one of the very important application. Wildlife forensics DNA barcode is very much used to prevent the illegal trade, because the site is listed, all these species are getting traded across the country, across the borders, where you can use DNA barcode. Next important application again in the food forensics. So food forensics, if you want to identify, see now that it's all the canned food items are very common, no? So if you want to identify the fraudulent ingredients, there will be false claims always in the label. So if you want to include, especially when there is some food poisoning is been happening, then definitely you need to identify the ingredients of the food. So this is the technology that is accepted by FDA. So it has been widely accepted across all the global agencies, and it's been extensively used in food forensics, and especially as Sir was mentioning about GMOs. See, GMOs are very widely cultivated in US, but when it comes to food, see, people, the public concerns, right? So already in British, there is a strict labeling, when GMOs are included in any of the packed goods, you are supposed to additionally label it with a DNA barcode. So that, see, as a consumer, we have every right, what to eat and what not to eat, right? So it is already been used extensively in food forensics. Next is it is for biosecurity. Biosecurity, I think it is mostly associated with biological weapons, right? And also whenever there is an infectious, say COVID. So it is again a biosecurity issue, right? But in a broad way, it is associated with the invasion of exotic plants and animals, biological invasion. That is also come under a biodiversity, biosecurity issue. So you see all these pest species, the top on the upper panel, you can see the plant species that is being invaded in the agriculture crops. It is causing productivity loss for the farmers, right? And it is affecting their livelihood security. Like that, we have the animal species, the pest species. The giant African snail is one of the worst invader in the world. Okay, and it is causing productivity loss for the farmers. So all these pest species, how it is coming, the major source of the biological pests are coming through the timber import. So in the quarantine being there is no expertise. See, most of these pest species are intercepted in various life cycle stages. Right, along with the timber, under the bark and all. See, so the quarantine officials, they don't have the expertise. We don't have any means by which you will be able to identify a biological pest from a life cycle stage, right? So these are the importance, like why it has to be, there should be some policy level recommendation which should include DNA barcode tool, so that, it is a very slow process of identification of the harmful pest. So by the time an official or quarantine means is identifying a pest, by then it must have spread beyond its ability to contain. So that is the worst scenario that is faced by many of the farmers in the country. They are facing productivity loss. So these are the interventions we should make at the government level. So now that again we are coming to timber forensics. So as I had already mentioned, we are using only two tools. One is like your human forensics, you are using STR profiling. Another one, since there are diverse timber tree species unlike human beings, we are using a DNA barcode to authenticate the species. So the STIR profiling, unlike human beings, we have to develop database for individual timber tree species which are highly traded. So human species, we know since the draft genome is known from 2003, we have the STRs that can individualize human beings. But for each timber tree species, say for teak, say for sandalwood, we have to develop that, and we have to develop the reference database. Like DNA barcode reference database, we have to develop an STR database for most of the timber tree species which are commercially traded, if you have to provide evidence or if you have to compare the seized log with that of the original stump, suspected original stump. So, as I had already mentioned, so the samples that are coming to us are not easy samples. It must have exposed to different seasons. It must have already attacked by termites. The termite exudate will be there. So getting DNA is the biggest challenge. And now that we have modified protocols for getting DNA even from shredded specimens. So, and then as you said, like once if you're able to get DNA, you have the barcode gene regions, right? In plants as well as in animals, it's the same workflow. And then once if you have the PCR product, you can go for Sanger sequencing. You can develop your barcode. So why there is a difference in color? It's not like your UPC barcodes are black and white, right? But here it has some 4 standard colors. Why there is 4 standard colors? Because all of us know, like, we have 4 building blocks for DNA, and it has some 4 standard ATCG colors. So, and the difference between among all of us is a slight difference in the order of arrangement of nucleotides, right? So that order we are trying to bring out by using DNA barcode. So DNA barcodes are developed from conserved gene regions, and we are just looking into the order of arrangement of nucleotides that is attached as a biological barcode for the species. So this was the DNA barcode database I was talking about. Like at KFRI, over the last 15 years of research, we were able to develop DNA barcode database for 43 timber tree species that has been published in Tree Biotech. And along with that, we have also developed an anatomy tool. So again, this is an integrated approach. Anatomy can identify only up to genus conclusively, very conclusively, because most of the species, the anatomical features are overlapping. So in that cases, you can supplement it with a barcode. See, if you cannot afford to for a barcode development always, you can think about microscopic means, but in case if you're facing a challenge, then you can supplement. In those instances, you can additionally supplement it with a barcode for species identification. So again, like this is the one of the first study in India where we identified the amount of market adulteration in sandalwood. This is published in Annals of Forest Science in 2014. So this was the first case study we did where we were able to identify the amount of market adulterants in sandalwood, and we even employed artificial intelligence platform to enhance the speed, precision, and the accuracy. So if you have some massive felling of some 300-400 trees where you have to compare stock with the stump, how will you do that? Manually it is not possible. So there you need a machine learning algorithms, and we developed that, and then now that we are practicing it for massive felling cases. So as I had mentioned, so this is the first of its kind of facility where we and major stakeholders of forest department, police department, and also like as I had explained about the forensic potential of DNA barcode, we had 2 sets of projects from Ministry of Ayush where we had actually developed DNA barcode database for Ayurvedic road drugs. So that is again another application, you know, like about the Ayurvedic products, the extent of adulteration that is happening, the deterioration of the quality of the Ayurvedic products. And so the Ministry of Ayush has given us a project in which we developed DNA barcode database for Ayurvedic raw drugs, and it has been very effective. We, as Sir was mentioning about the industry collaboration, now that we are associating with 4 Ayurvedic drug manufacturing units, where if they want to confirm, and especially this is for value addition of their product, many of the Ayurvedic products are having export value. So if they are going for high-end marketing, and if they want to enhance the value, if they attach the DNA authenticated label, then that can be used as a value-added product. So we are associating with 5 Ayurvedic drug manufacturing units after the development of DNA barcode reference database in Ayurvedic raw drug with financial support from Ministry of Ayush. So as I had mentioned, the second tool is about the STR profiling, like we do in human forensics for parentage analysis. So here actually we are using STR analysis for log to stump comparison. And see STR, so SSR, so microsatellites, as I had mentioned, humans, we know like after draft genome is known, we have the set of VNTRs or STRs which can actually discriminate individual human beings. But for this timber, we don't have any means, we were not having any database. So the first step when we started off this concept was to develop DNA reference database, especially the STR, SSR reference database. So as you know, like sandalwood, it's mostly, most of the populations are restricted to South India. And we know like earlier there was a monopoly, nobody was growing sandalwood. Why? There was a government monopoly by which regulate the trade of sandalwood. You cannot, you can grow sandalwood, but you cannot sell it off. So there is no point in investing money for sandalwood plantation, right? But because now that since the species is vulnerable, now that there is a policy shift by which the government is encouraged to grow sandalwood plantation. So every year now that 600 hectares, so the farmers can grow, they can harvest, they can sell the timber, and they can get profit. So sandalwood plantations are also getting, but the main problem is the security issue. Like once it is grown, once it is 15 years, that is maturity period, then definitely you know like what all things are happening. So there also most of the cases, 99% of the cases that are coming to us is of sandalwood smuggling. One for species identification, one for, now that it is not from private planters, it is not from homestead. Some are from homestead, but mostly from, the province like Marayur. Marayur, you must have seen the sandalwood. It's one of the best province in India where massive felling is happening. Even though like there are many preventive measures including radiometry, but the felling is happening in a very rampant rate. So what we did is like, these are different newspaper reports, and there was recently a very strong Supreme Court ruling in which they were able to prosecute the offenders. Why? Because there was no scientific evidence. So there was a strict ruling from Supreme Court of India very recently. And what we did is like in 2019, we developed, we partnered with IFGTB, Institute of Forest Genetic Cities and ICFR Institute. We partnered with Dr. Madhumita Das Gupta and myself. We developed a draft genome of sandalwood fir. So it's a very small genome. You know, like it's only 286 MB. So once if you have the draft genome, then definitely you can mine out your STRs. Unlike human beings, like you have to develop for individually for each tree. So once after development of the draft genome, after the development of the genome by SSRs, we have to do STRAF. That is a very important bioinformatic analysis for fine-tuning the STRs of forensic potential in any tree species of interest. So these are the SSR genotyping patterns, and this is the way how we are identifying, or we are comparing the log to stump. See, these are the different allelic patterns of different SSRs, and the same color is all this left panel represents that of a source wood. I mean the stump. Okay, and the right panel represents the seized wood, and the same color represents the origin of that confirms the origin of the seized wood from the original stem. So this is the way we are doing the STR profiling for log to stump comparison. And similarly for teak also, like both these are supported by Department of Biotechnology, Government of India. We have a multi-institutionary project along with CPMB, Center for Plant Molecular Biology, Hyderabad also. So we developed a draft genome for teak first, as I had mentioned, we developed the genome-wide SSRs, then we subjected the genome-wide SSRs to stress analysis. Now that we have a small set of STRs which can be used for forensic applications in teak. So again, the draft genome of teak is of 317 MB, and we have identified the STR markers which have the very good forensic potential. So and when it comes to Indian rosewood, we know like there are more than 200 species, but it is again in Kerala also we witnessed a massive felling case where more than 300. When we visited the field, it's very sad to see the logs. Like, it's such a royal tree. I mean, the color and everything, it's like more than 300. Age also we estimated, and it's all the logs are 205 logs. 205 logs, all are of about 300 year. So all these are cut from the reserve forest. There is a very strong mafia involved. So when it comes to the UNODC report, you can see, I don't know whether it is visible, but the rosewood, 18% of the illegal trade is happening in Indian rosewood, and that is along with the world wildlife crime. So rosewood is the most hunted tree among the tropical timber tree species, and this is a World Wildlife Crime Report. You can see the upper graph, the blue graph. That is again showing an upward trend over about, like see the elephant ivory, pangolin scales. It is showing the illegal offenses that is happening in the country is much above all these. So that is across the globe. UNODC and World Crime Report is a global comparison. And you see the extreme IUCN comparison where they have listed the across countries the amount of offense is happening. So India, again, it is 25% of the illegal trade is happening in Indian rosewood. But unfortunately, we don't have any genome in place for. So what we did is like and these are the different newspaper reports, and this is the middle one is of the Wayanad, the Mutil felling and the log scissors. And so we proposed in 2023, we proposed a project to Department of Science and Technology, Government of India, and I think it's the first NRF. After the NRF is in place, we were able to get this project. So now that the draft genome is developed, and we are doing the analysis, and we are developing the genome-wide SSRs, and we have to see in all these cases, in the case of sandalwood, in the case of teakwood, what we did is like after the development of genome-wide SSRs, we have sampled individuals across India, and we developed a database. Minimum 450 samples in all the timber tree species we have genotyped. And then only you can create the reference database. So if you have a reference database, only you can do the straff analysis. Straff analysis, if it has to identify the STR markers with forensic potential, then definitely you need to have a database. So, and again, now that we have a ITTO, International ITTO project, again for an integrated approach, in which we are using an integrated approach. As I had mentioned earlier, we are using isotopic profiles, we are using FTIR profiles, and this is an integrated approach. Thankfully, I'm in time. So this is the last slide. So coming to the future directions, like as we know, like now that KFRA is the only one institute, we went and made a presentation before the IG, Minister of Environment and Forest, and he was the one who, like now that we are trying to make collaborations across the country. We have 8 ICFRE research institutes in various states. So now that we are partnering, because it's very difficult to deal with, I'm getting phone calls from Northeast, I'm getting phone calls from Karnataka, Telangana for attending to the timber theft offences in their state. But see, the most tragic part is like whenever the trial is happening, if it is a very high profile case, then the officer responsible for have to legally present before the court. So that's very difficult to appear in the court each time when the trial is happening. So we have enough and more offences happening in our state itself. So we are restricting our activity to the state, and we are going to be approved as a nodal center for timber forensics for the state of Kerala. Likewise, we are partnering with, after the patent is done, then we will be again, as sir was mentioning, like we will be sharing the DNA extraction protocols with most of the ICFRE institutes, so that together we will be able to combat illegal filling, and we should have a very stringent mechanism by which we would be able to prosecute all these traders. So not only in the countries, it's very essential, like across the borders also this illegal trade is happening. So there should be a global collaboration. Globally, there should be a concerted. That's why our ITO project stands for ITTO project. Japan stands for that. So we are developing, we are partnering with the different countries. This is for especially for rosewood. So all the rosewood growing countries are coming together for developing a global database. So if you have to develop a global database, there should be a harmony among the forensic tools that is being used. And the most, and I think the essential part is the capacity building program. So awareness programs for the quarantine officers, for various stakeholders, whoever is involved. Now that many timberwood industries are coming to us, contractors are coming to us before they go for the construction, they would like to confirm the authenticity of wood. So we are doing consultancy project for the different timberwood industries so that they will be able to use the correct, not the adulterated exact wood for the construction purpose, especially government departments like PWD, where we are doing the DNA testing for species authentication. So with that, I think I'll conclude my lecture. So I would like to thank our former directors, our present director, my colleagues, Dr. Donald James, and who is helping me with genomic part, Dr. Sandeep, who is also a principal scientist, who is working into the isotopic tools and spectral signatures. My students at PhD students at various stages who were part of these timber forensic activities, different funding agencies like Department of Science and Technology. They were very supportive. Department of Biotechnology, I think 4 projects we operated for the development of the reference database to DBT programs, and Kerala Forest Department. They were the partners in many of the projects, and now that with the head of the forest recommendation only, like it has gone to the government for approval of the center. And these are the various international collaborators. Right now we have a program with ITTO, we have a program for rosewood, and we have a program with World Forest ID where we are developing a global reference database for teak. So we are also partnering with them. So thank you. Yeah, I thank the organizers of 32nd Swadeshi Congress, especially Professor, Dr. Jasmine, who we were in touch with, and I thank especially my mentor and teacher, Dr. G.M. Nair, sir. So I'm really grateful to him because I think Dr. Jasmine will also see. Still, I could recall, the PG and MPhil days about his lectures in biotechnology. It was truly inspiring, and I think that laid the foundation. So but I think definitely she will agree with me also, see what we have achieved till date. I'm grateful to you, sir. So the first time that was long back in 90s, 96, 97, that period. First time when we, see that time like your first generation markers like the RIPD, RFLP, all these things had just come. So first time when we were able to cast one gel, we were just taking the sensor, we were able to get the gel, our isozymes and all these things. So that was the very foundation for us. So I'm really indebted to him in multiple ways. Thank you, sir. Thank you so much.
Your talk is very impressive. Especially it is new most of the botany students regarding the tracing the illegal timbers. You might have heard about the film Pushpa. How that it is $1 million international business, timber illegal trading in India.
Yes.
So that I think that Pushpa director would have seen this molecular tracing. You might keep it in your scene. That is a very impressive because that will spread to the common people.
Correct, sir.
So this tool identified for detecting the illegal timber, now it can be taken into a court for validating it.
Yes, yeah.
So madam described it very nicely. I request 1 or 2 questions from the young participants to the madam.
You can also refer to the KFRA website. We have an attachment program. We have PhD programs of different universities, not for PhD. And even we have internship programs. We have MSc attachment programs. So I think now that it is compulsory in most of the semesters. So if you want to come in as an intern, you can just refer to the website. All the details is being given. So just 2-3 months you can be attached to the facilities and you can be exposed to all these things, so that can lay some foundation for you to take up your research career further. And as sir was mentioning, like Cliff, we have a very good central instrumentation facility with, we are having paid services. We have all the high-end analytical equipment. So that is also been, all the details are given in the website. So we are also doing paid services for DNA barcoding to, not for academicians, because academicians, then it will go to a research project. But when it comes to for different, the stakeholders are majorly judiciary, forest departments, PWD, and different industries, Ayurvedic road drug industry. So all these, but if you want to develop one or two barcodes, see, sometimes some students used to have, madam, I would like to develop a barcode for cinnamon. There was one request. So like that, one or two requests we used to entertain. So you can look into the website for the details and get in.
I have one doubt on. See, can you try any image capturing from the cut of the timber? That would be some species level.
Sir, that's.
Find out any differences on.
Yes, sir, yeah. See, the thing is like that's why we are using now, along with the genomic tools, we are using an integrated approach where you are using spectral signatures. So FTR spectra is exclusively for that. So there are some minor, so that will capture the surface features. So along with the genomic tools, like, see, as I had already mentioned, whenever we have some complications with regard to the yield of DNA or quality of DNA where you cannot proceed with further, you will be using, so both these can be an integrated approach. So that is, definitely it will be more efficient enough.
Definitely it will be very useful to the government.
Even we have tomographies nowadays where you can scan and you can estimate the hardwood content. So that much advanced the technology is. You don't have to, sacrifice a timber for estimating the hardwood content as earlier days. See, you can have, just like your CT scanning, you have a tomography scanning where you can estimate, see, sandalwood and all, like if people are establishing plantation, if they want to know like whether the felling cycle has reached, how much yield will be there, all these estimations can be done by tomography now. So that much advanced our technology is.
Any doubt?
Yeah, you said you were using the conserved regions, especially like as markers.
Hmm.
So when you do this stock,
Reference database? No.
when you are trying to
Okay.
match the stock with the what is
Stump.
stump, do you face any difficulty like with individual matching?
See, the markers, there are two types of tools. No, DNA barcode also we have diverse gene markers. That is for species authentication. But when it comes to log to stump comparison, it is STR profiling. Like you have a set of STRs for paternity testing in human beings, we have a set of STRs for each of the timber tree species. And these STRs are selected based on a reference database. So what will we do is, like, once the draft genome is in place, we will mine out the genome-wide STRs. Okay, and suppose you have, and you will be doing always in, see, at least minimum 6 samples you will be using. One you will be going for high depth intense genome sequencing, and rest you will be going for low depth. So you will be able to identify in silico polymorphism from the database itself. So from the in silico polymorphism, you will be selecting, say, some 50 markers, 50 STRs.
Able to perfectly match the
Yeah, that's what I'm coming to that. So with those 50 STRs, you will be doing genotyping of all the individuals across different accession in the country. Say for sandalwood, we genotyped 450 individuals across different distribution zone with the 50 primers. Okay, 50 STRs. And then you will apply your straff analysis. So straff analysis will scale it down to some 10. And those 10 does have the ability to individually differentiate. If you have an STR marker which can individually differentiate the wood only, you can use for log stump comparison. So that is the way we are. So that's why it took so many years.
My question was, have you come across cases where you found difficulty in matching?
Definitely, see, for the Mutil case itself, see, if there is a massive felling is happening where you have close to 200 seized logs and you have 105 stumps to match. See, so 200 profile with 10 markers, you can just imagine 200 into 10. 2000 DNA profiles you have with your 1,000 stump. So definitely then in that case we used mycorrhiza, and in many 24 trees we were not able to get good quality DNA, that failed. So it doesn't mean that in all the cases we receive and those samples were in the exposed to sun and rain for 2 years. Then only we would, and it was also attacked by termite. Okay, so in some cases we do face difficulties.
So this stump was seized from somebody.
No, the stump will be, see, stock, yes, exactly. So this particular log was got from Perimbavur, truckload full of rosewood logs, huge dimension. That's why I said all the logs were about 300-year-old, and it was seized and it was in one of the depots in Sultan Batheri. And then from there only we collected the samples. So,
Any other doubt?
Actually, I always wondered about the sandalwood theft because in our university, especially in our botanical garden, we are having some sandalwood trees, young
okay.
ones. Older ones are disappeared. Uh, so even actually,
Have you registered the case?
no, actually we know that they were disappeared
Okay.
because when we came to the university, we could find that missing these things, what you can say, the remnants. So the thing is, at that time I was not knowing about case filing and all that.
Okay.
But now, when the younger ones are growing, we are always wondering how to secure this. Because in Kasargod, it is a written rule that if you have a tree, when it is matured, 100% sure it will be gone. Whatever you do, it will go. So, if there is some database where we give our leaf samples now itself, a deposit and create some database that someday somebody is stealing, that can be that.
But there should be seizure also along with that. If you are not able to seize the logged wood,
So in case somewhere if it is seized and if they check, they will get to know that
definitely.
it was a theft case.
Yes.
But if some system is there, then there will be a fear in the mind that no, if at all they are caught, then they can backtrack.
Now also that fear exists because nowadays, like for I said no, over the 2 years, 49 cases we have submitted evidence. In most of the cases, the illegal traders were prosecuted. Earlier, the instance was like when the trials are happening, no, there is no scientific evidence and the people will walk out scot-free. So that will encourage them to, because this is equivalent to your narcotic trade. See, sandalwood, 1 kg, it is how much the cost, it's coming to 15,000, 12, good quality sandalwood, 15,000 to 12,000 to 15,000 per kg. See, so that is the main attraction towards this, and there was no conviction happening so far. But now the situation has changed. Whatever the theft is happening, samples are coming to us. And now recently, like last month, there was a sandalwood theft in CWRDN campus in Calicut. So the sample has come to us, but there should be seizure. And as you said, like once if you create the database, we can safeguard the database, but that doesn't mean that your trees are safe. Always that, see, the crime can happen, but
Now, if suppose if a theft is happening, what should we do? Should we?
we have to register the case first with police, yeah. He's,
From the student side? So not by the faculty members.
restricted.
So I request some of the doubt from the students. Actually, the topic is very interesting. That's why the faculty members are very much involved and interacting with you. So otherwise, you can talk with the madam when you are.
You can even visit KFRA because we do, and we have an extension training department, and we do entertain student visits across universities from different, even Northeast students are coming to us. So we have many live collections, museums, different, very good and high-end infrastructure with all the state-of-art laboratories, and we have a commercial production unit in plant tissue culture. So you are invited to, welcome to visit our institute. You can just look into the website if there is a genuine interest.
I request
So.
my co-chair, Professor Nagarajan, to propose the vote of thanks in this session.
President, madam. To all of you. So now we have come to the end of this session.
Thank you, sir. I request co-chair Professor M. Nagarajan, sir, to hand over the memento to Dr. Suma Arundev as a token of our gratitude.
We will now commence the oral presentation session on the theme of horticulture and forestry. We welcome our judges Dr. Ginny Anthony, Assistant Professor, Department of Plant Science, Central University of Kerala, and Dr. Jasmine M. Shah, Assistant Professor, Department of